Mm-hmm. Muhammad, boleh cuma kerja. My first is life.
Okay, good evening to all. So I'm welcoming Dr. Abdul Samad sir for giving this guest lecture. So I'm welcoming all the participants as well as our students and today's guest for this guest lecture on how to conduct a systematic review, which is going to be delivered by Dr. Abdul Samad. And so before going to start the session, so I'm inviting Tony to give a brief profile of our chief guest today. Yes, Tony.
Thank you, sir. Good evening to one and all.
Good evening.
Yes, sir. It's my privilege to read out the profile of Dr. Abdul Samad Ji. Abdul Samad Ahmad Mokbal Al Salahi, sir, is a pharmacist and a member of the MNE Pharmacist Syndicate. He is a lecturer at the Faculty of Pharmacy, Sana University, Yemen, from 2002 to 2010. He also completed his PhD in pharmacology from the Faculty of Medicine, University of Malaya, Malaysia in 2019. He holds a bachelor degree in pharmacy from the Faculty of Pharmacy, Sana University, Yemen, with a 3.4 CGPA with Honorary Order of Merit in 1998. He is also Master of Molecular Medicine from the Faculty of Medicine, University of Malaya, Malaysia in 2013, and he has 12 publications with the H-index of 7. He has been serving as a reviewer of a reputed BMC complementary and alternative medicine, as well as in order to add something, sir has also a number of memberships and as well as social activities. He is a president of MNE Student Society in University of Malaya, Malaysia in 2015 to 16. He is also a president of Postgraduate Students Club, Department of Pharmacology, Faculty of Medicine, University of Malaya in 2016 to 17. He is also a member of academic staff, Faculty of Pharmacy, Sana University, Yemen since 2018. So it's our pleasure to invite you, sir, and thank you for accepting to act as a speaker in this guest lecture. Thank you so much, sir. We welcome you on behalf of our Department of Pharmacology, CHALAPATHI Institute of Pharmaceutical Sciences, and we request you to proceed the session, sir. Thank you.
Thank you so much for CHALAPATHI Institute of Pharmaceutical Science, and thank you for the organizers. Thank you for your invitation. We will start now. Now we can start with the today it is my pleasure to introduce my seminar about how to conduct a systematic review. Let us start. The scope of this seminar was prepared to be from the viewpoint of pharmacology, because most of the attendants are either pharmacists or who are interested in the medical field. This to develop the methodology in to develop a methodology in a simple applied manner also, and to address the most common problem during conducting a systematic review. First, we can start with the type of evidence. You know that there is two types of evidence, either primary evidence that comes from the primary literature, such as original research articles, or a secondary evidence that comes from the secondary literature, such as books and reviews. You know that there is a huge number of medical biomedical studies in the literature regarding a particular problem of interest or intervention, and the available studies are heterogeneous because they contain either clinical trials or pre-clinical trials. So this huge number needs to leads to an increasing demand to summarize and apprise the evidence. So there is always a tool to do that, to apprise and to summarize and apprise the evidence from the literature. The first one is to gather the records from the database, then to select the records that are related to the health problem of interest. Then to apprise the related or the selected studies or records, and then to collect the data, summarize the data, then to interpret the data. The types of reviews in general, as a general term, overviews are a term that is called for the several type of reviews, like narrative reviews, scoping reviews, systematic reviews, and even meta-analysis. But it goes further to be restricted for systematic review. Systematic review is different because it focuses on only evidence that follow evidence throughout or across all the included studies. Then meta-analysis come later after doing the systematic review to gather the to combine the evidence in the form of estimator, to take the estimator like standard deviation, mean and standard deviation, or hazard ratio, risk ratio, odd ratio, and to combine together to see or to evaluate the size effect. Then we can go further to do individual data for patient meta-analysis for individual data in each group, either in the experimental or in the control group. What is a systematic review? A systematic review, it is an evidence-based review that formulates a research question. This is very important. The research question is very important in the systematic review because it is the guide of the systematic review, and it is fundamental to formulate all the other methodologies, methodologies like search methodology, selection of studies, and also eligibility criteria, and so on. Also, the systematic review following a systematic and reproducible methods to identify, select, and critically apprise all the relevant research, and also to collect and analyze data from the data also that are included in the review. There are several features for the systematic review which make is advantageous for over the other type of reviews, because it follows a transparent, strict, and comprehensive systematic methodology. So for this feature, systematic review is always reproducible, updatable, and unbiased. This is the most important advantage of systematic review. There is a question, why do we conduct a systematic review? There is two common reasons to conduct a systematic review. The first one is when there is conflicting primary evidence in the literature. Sometimes some studies, for example, said that this intervention is effective, while the other studies reported that it is non-effective. So there is a conflicting results. We can do systematic review to solve this conflict. The second case, there is accumulated primary evidence in the literature, but have not yet systematically outlined. So in both cases, either in case of solving the conflict or doing the systematic review for the first time, this helps the policymakers to make a decision, or even a student to support his research proposal. The types of systematic reviews for clinical studies, it may be interventional, includes randomized control trials and quasi-experimental design, or it may be observational, includes cohort cross-sectional case control studies, or it may be diagnostic, to include diagnostic test accuracy studies. The systematic review for clinical, preclinical studies, you can conduct animal-based systematic review for animal-based studies, but the evidence from animal-based studies are not generalizable to humans. But also, it can direct the future research to conduct clinical trials, for example. Whereas the systematic review for the evidence from cell-based studies cannot stand by itself. but it can combine with the evidence from animal-based studies. There is some points to pay attention for before conducting the systematic review, because systematic review, it needs longer than 9 months. It is time consuming, and also it needs huge efforts for reading, writing, many things. Also, it needs resources, like you need database, you need teamwork, you need also softwares, you need also money, for example, to translate the manuscripts written in Chinese, for example, into English, or vice versa. So the first stage in the systematic review, first to rationale the systematic review. Why you conduct a systematic review? The first one, this our topic that will be selected as an example in our systematic review. Clinical efficacy of givatinib to improve the overall survival of patients with non-small cell lung cancer compared to placebos and standard chemotherapies in randomized controlled trials. This is an example to be discussed within throughout the slides of this seminar. The first one in rationale of the systematic review is to analyze the current gap in the literature. You can check when you intend or going to conduct a systematic review, you have a certain topic. For example, that's the example that we mentioned before. You should first check the literature, like Bruce Bureau and Cochrane. Bruce Bureau and Cochrane are very important database to be checked first, because most of the prospective protocols are registered on two platforms, particularly or more commonly Bruce Bureau. This give you idea about if there is ongoing systematic review or a completed systematic review. And also you can check database of abstracts of reviews of effects, or Google Scholar, or PubMed, or another database, to get sure that there is no ongoing systematic review, similar ongoing systematic review or completed systematic review. So you have three cases in this case. There is either similar ongoing systematic review or completed systematic review. In this case, you can change your topic, because you will repeat a work that is done before. The second case, no current similar ongoing or completed systematic review. Okay, you can go ahead here. There is new evidence to update a similar completed systematic review. For example, there is a systematic review about a certain topic from 2010 up to 2015. And published. This systematic review is published, but there is new studies come after that. So you can update the systematic review by combining the whole result from 2010 up to 2020. Okay, there is also a special case if there is, you can check if there is enough studies, because sometimes enough studies are not informative. Suppose that you can proceed also, you can proceed systematic review for 2 studies, but it will be not informative, because sometimes there is a heterogeneity in the results. You can see that some studies said that one study said the glycemic activity or the hypoglycemic activity of glibenclamide is effective. The other said it is not effective. So there is a conflict, and you cannot get informative conclusion. Sometimes there is publication bias, because you include in your studies 2 studies that have positive results, while there is no studies that have negative results. After that, analyze the gap, you can formulate a specific research question. This is very important. You cannot conduct your systematic review without formulating a research question. The research question is fundamental to develop the research strategy, selection of studies to be included, and type of data to be collected. Here I give an example here. as an example, how to formulate your research question. For example, in randomized control trials, does the first line monotherapy with gefitinib improve overall survival of patients with non-small cell lung cancer as compared to placebos and standard therapies? You can see that randomized control, you should, here you should formulate your research question according to BICOS. BICOS mean B for population, I for intervention, C for comparators controls, also O for outcome, and S for study design. These elements of BICOS should be contained in your research question. Here, research randomized control trials is the study design. The intervention is gefitinib. The overall survival is the outcome. Population are the patients, humans. Non-small cell lung cancer is the health problem of interest. And placebos and standard chemotherapies are the comparators. So this is our research question. So we can formulate the research strategy and selection of studies and also data collection according to the research question. After you formulate your research question, you should provide a specific rational background. You can formulate it as problem statement. You can provide background for the intervention and the health problem of interest. Also, you should review the related works and you should state the current gap in the literature and state the significance and state the hypothesis and objectives. This for the background. The other element is the, make specific relevant, provide a specific title. Yeah, even that's the title should be formulated according to the research question. I give here example, efficacy of gefitinib on the survival of patients with non-small cell lung cancer in randomized control trials as systematic review. Here, this is the intervention, the outcome. Patients are the population, non-small cell lung cancer is the problem health of interest, and the randomized control trials are the study design. Systematic review should be referred here in this case because you can because to distinguish your study from the randomized control trials, also you, for the narrative reviews, scoping reviews, and so on. The second stage is to develop and optimize methodology. The methodology should be prepared according to PRISMA. The first element in the methodology is to develop a search strategy. The search strategy is the cornerstone of the systematic review methodology because it allows to gather the records from the literature review, and also it makes the methodology reproducible. The search methodology consists of three elements: the keywords, source of information, and filters. We will discuss everything later. Okay, first, how to develop keywords. The keywords syntax should be constructed in response to the research question. For example, you can use intervention against health problem of interest, or intervention against outcome, or intervention against study design. These are the examples. For example, for intervention against health problem of interest, there is gefitinib against non-small cell lung cancer, and gefitinib against overall survival. This for the outcome. And gefitinib against randomized control trials. These for the study design. We construct them, the keywords, according to the research question. You can also use Boolean operators to formulate the syntax of keywords using and or. They should be capitalized. We can see with or operator, using OR operator let you broaden search, very wide search, broad search. Because, for example, if we construct this keyword gefitinib or non-small cell lung cancer, In this case, when searching in PubMed, this will give you studies that contain givatinib alone and studies that contain non-small cell lung cancer alone, and also give both studies that contain both givatinib and non-small cell lung cancer. When searching in PubMed using these keywords, the results is very huge, 59,745. Let us try to how it will be with AND operator. With AND operator using givatinib and non-small cell lung cancer, only this make narrow your search from the database. How? Because it will give you the studies that contain givatinib and non-small cell lung cancer, not givatinib alone or non-small, both. So in this case, when searching in PubMed, the number of the results are 15 as compared to 59,000. Also, you can construct or build your keywords using medical subject heading mesh of NCBI. But in this case, there is a taxonomy inside the database. For example, when I try to search with givatinib and also I search with non-cell lung cancer, it suggests the mesh suggests to use givatinib and carcinoma non-cell lung cancer because this is stored in the mesh. Okay, when searching in PubMed, the number of the results were 2,149. So in this case, you should optimize your and validate your keywords. For example, here, try a pilot search strategy first to construct the keywords using the Boolean operators or mesh as do you want in this case, but you should optimize them, tailor the keyword syntax to the threshold of a comprehensiveness, comprehensive retrieval of the related records. And also you should consider the different spellings like randomized and randomized. Also you should consider the different synonyms, tumor and cancer. You should also consider different names of the same intervention, for example, paracetamol and acetaminophen. Also, you can consider keywords from the related published papers and systematic reviews, meta-analysis, or narrative reviews. You should also consider seeking help from experts to validate and optimize your keywords. The second element in the search methodology is source of information. We have two types of literature here. We have published literature and we have gray literature. In the published literature, this focused on the database that stored the published studies, like PubMed, Scopus, Science Direct. But there is a drawback in the published literature, which is publication bias. Publication bias because the published literature tends to accept studies that have positive results and reject the studies that have negative results. This not always, but in most cases. So gray literature will give you the studies that have not published elsewhere. This gray literature embraces studies like conference proceedings in many cases, and thesis and dissertations. There is database for gray literature online or are available online, like OpenGray, Trib Medical Database, Midner, and Brookquest that is a database for thesis and dissertation. You can also consider contacting known researchers in your field because he knows a lot about the studies that are published or unpublished. You should also visit extra source of information. These to broaden your search to make a comprehensive search for your records. You can visit online libraries like Nature, Elsevier, Gestage, and so on. You can also visit registry platforms for clinical trials, such as Clinical Trials Registry in India, for example, Netherlands National Trial Register, to give you the recruitment status of the studies. Means that either it is completed or it is ongoing, for like that. You can also search Google Scholar. Google Scholar here is very important because it gives you the works that are cited for these studies, and also you can visit also list of references, because it may contain studies that are not published, or even published but they don't exist in the database. Okay, the third element is the filters. They are identifiers to refine your search strategy. For example, you should make a filter for time frame. You may include studies only from 2010 up to 2020, or as do you like. You can also refine the results by language. You can refine by country, by type of published documents, by study design. This also you can check the search strategy by using BRESS. There is BRESS checklist or PDF for checking your search strategy if it is optimized or not. But you should focus in this case, in case of search strategy, you should have a teamwork consisting of 2 independent investigators and a third person to solve the discrepancies that arise during work by the 2 independent investigators. The second step in the methodology is the study selection. The study selection, there is 2 types of selection, either primary selection or secondary selection. In the primary selection, you focused on selecting the related original research studies. This because after implementing the search strategy, you have a huge number of records or studies. So you should first, after sending these studies to EndNote, you should remove the duplicates by EndNote. And then, because you are focusing on selecting the related original research articles, you should exclude the secondary literature like books, reviews, guidelines, and also miscellaneous like protocols and so on. In this case, in case of primary selection, you should screen the records by title and abstracts. Once you have finished the primary selection, you select the original research articles, you can go to the secondary selection. The secondary selection is more specific than the primary because it depends on the eligibility criteria, inclusion and exclusion criteria, which should be formulated in response to the research question. In this case, we will screen the records by faulty text. The faulty text, you can get it from several sources. Also, in this stage or in this step, we need a teamwork, the same teamwork for search strategy, 2 independent investigators and 1 and third person to solve discrepancies. This is an example for the inclusion and exclusion criteria, which were tailored according to the research question. First, study design. In our research question, we include randomized control trials with its characteristics. We will exclude preclinical studies and observational studies. And the same thing, you can do it with all the elements of the eligibility criteria. The third step in developing the methodology for the systematic review is the appraisal of the eligible studies. This very sensitive step, because you will assess the quality of the included studies. Because many studies or studies, there is bias in the studies. The bias will either go to overestimate the size effect or underestimate the size effect. This is very time consuming because you should read everything in the paper, and you should be frank and transparent during the appraisal. The source, the assessment of the risk of bias should be implemented at the level of studies, and you should assess the bias across the studies and within each study. You can use Refman software of Cochrane for making appraisal. The source of bias in the randomized control trials according to the Rob-2 tool of Cochrane. The first one is the selection bias. In all type of bias, the bias that is committed during studies is subjected to the assessor preference. So there is selection bias. In selection bias, there is no random sequence generation, no randomization method. And also the allocation of the patients in the interventional group and also in the control group conciled by the assessor and the participants. Also, there is another type of bias, performance bias, which there is no double blind for the assessors and the participants. There is also a detection bias, which is due to there is no blinding for the assessors when measuring the outcome. Attrition bias, not adequately address the missing data, withdrawals of the patients if there is death or discontinuation of the patients or the participants. It is called also incomplete data. Attrition bias is called also incomplete data. There is also reporting bias. Reporting bias, it's selective reporting for the outcomes. Either to you have, for example, in your objective, a primary outcome and a secondary outcome. But during reporting, you only report the primary outcome or the secondary outcome. So there is selective reporting. This is reporting bias. There is also other source of bias, like for example, this depends to compare the content of the manuscript with the prospective protocol in case of randomized control trials. For example, in the prospective protocol, the investigators that said that the primary outcome was the blood glucose level, and the secondary outcome is the insulin level. When he reporting the results, he makes switching for the outcomes. Or sometimes there is funder bias because the sponsorship of the trial is a pharmaceutical company. Now we will go to the data collection and extraction. The data collection, you should state from where you will extract the data, from text tables and or graphs. Also, you should state how you will address the missing data in each included study. You should contact the corresponding authors, the corresponding author of the manuscript. Also, you can use to collect data, you should use Excel spreadsheet or systematic review software. Usually we prefer Refman because it is free and also it is advanced. But there is special points in these particular points in during data collection is should be blinded. The data collection should be blinded. How blinded? You should disclose any identity of the authors because the perception bias. Because sometimes suppose that this study is conducted, for example, in a certain country, and the authors have hate this country, or said he is not sure about this. So there is perception bias. Also, you need also teamwork for two independent investigators and a third investigator to solve the discrepancies. The type of the data that will be collected will be in response to the research question. For example, study design, you can extract all the data that related to the study design for interventional model like parallel or crossover, the randomization, and also the method of masking, the settings, it is global or multi-center. Even that you should give the number, the ID of the prospective protocol, and also the recruitment status of the randomized control trials and the follow-up. For population, also the same. There's many, you can extract all the characters that are related to the domains like the study design, the population, problem, everything according to the research question. The study characteristics of the included studies, you should summarize the characteristics with citations. For example, for study design, you should mention that how many randomized control trials, how many parallel, which were parallel design, which are crossover design, and also the population, how many population, this the included studies, how many the sample size that's covered by the whole studies in the control and in the experimental group. Also the characteristics of the intervention and comparator, and you should set a table like this to summarize and extract the data inside this table. There is the data sciences. In our example for systematic review, only we will proceed qualitative literature, narrative. You should summarize your data to synthesize a qualitative literature. Sometimes you may combine it with quantitative literature meta-analysis. If you plan to implement meta-analysis, or a mix of them. In the qualitative senses, you can also present the collected data at the level of outcomes. And also you should critique and show the drawbacks and limitation of each of each study. You should consider how to address heterogeneity of the data across the included studies. Then when you develop your systematic review and the methodology, you should register your systematic review in Prospero. That's all, and thank you.
Yeah, thank you so much, sir. Hello.
Hello, yes, yes.
Yeah, thank you so much, sir, for the thing what you have shared. The complete things on all the prospective research, as well as a complete systematic study, and blinded as well as double blinded. Most
Yes.
of the things, because our students have also the same things in their syllabus.
Yeah.
We have shared much more useful content where it will be, our students will be get more benefited by these things, by these type of things, sir.
Yeah, thank you, thank you.
Thank you so much, sir. Sir, tell me, sir.
Thank you. Thank you so much.
Thank you, sir. And students, as well as other participants, if you have any queries, you please feel free to ask our speaker.
So, okay.
So we welcome all the participants. If you have any queries, you can ask our today's speaker. Thank you.
Okay, thank you. Thank you so much.
Sir, I have one small query, sir.
Okay.
Sir, regarding the gray literature, what you have said regarding gray literature.
Gray literature, yeah. What
Yes,
about?
yes. How can it be useful for our publications? How can the information in gray literature can be useful in order to publish our things?
For your systematic review, you mean?
Yeah, yeah.
Yeah, because the published literature focus on the studies that have positive results. So gray literature, you can get benefits to include the studies that have negative results. So this you will get the direction of the systematic the results. If the majority of studies that there is effective results, it will be okay. The final conclusion will be this intervention is effective.
Right. Thank you, sir.
Thank you. Okay.
Right. Amir Asad, sir, do you have anything to ask?
Oh, it's okay.
Amir Asad ji, do you have? I think you have unmuted. Do you have anything to ask?
Oh, it's okay.
ينادوني عاد انا مانيش داري اشوف ينادي
Right. Anyhow, sir, I think the participants, they don't have any queries till now.
It's okay. Thank you.
It also quotes the thing, whatever you have shared is very clear to them, I think so.
It's okay.
Right. Thank you so much, sir. Thank you for accepting our guest lecture.
Thank you so much for your hospitality.
Thank you, sir.
Thank you.
Thank you so much, sir. And
Okay.
we invite these type of guest lectures in our future also, sir.
Inshallah. Maybe. Okay. Thank you.
Thank you, sir. And my dear participants, you can leave the room now. Thank you so much.
Okay.
Sir, 1 minute, sir.
Okay.
In the chat box, Somaya posted that he has some question to ask.
Yeah, in the you can read it because I can't go to the.
Right. Dr. Somaya, do you have any question to ask? Yes.
Yeah, I just have one question about the question. كم
Hello, excuse me, Amira sir, please mute your mic. Amira sir. Yeah, thank you. You can ask Dr. Somaya. You can ask now. Thank you.
Okay. Hi, and thanks a lot for Dr. Abdul Samad for this informative workshop. I just I have one question about the formulated of the question of the research or about the review systematic. When he mentioned to be cause, is this only applicable for science or can be also applicable for social science? That's all. Thanks.
So for because we have in our field of study for pharmacology, always there is intervention. So this more specific for the medical field or for the interventional studies. But you can still have the type of study design, and also you have a problem. You have also population. You have also comparator. You have outcome to be measured. So you can you can make it compatible to your topic. Even that's in social science.
All right, thanks. Thanks a lot.
Okay.
Right, thank you, Dr. Somaya, for the question. And thank you, Dr. Abdul Samad, for the today.
Okay.
Thank you so much, sir. And dear participants, you can leave the room now. Thank you so much. Thank you for your participation.
Okay, thank you.
Okay, thank you, Abdul Samad.
Thank you, bro. Thank you, bro.